Okay. Hi, everyone. Thank you very much for coming today. First, Keiko, thank you very much for your excellent introduction. I think this is the best introduction in my research career. Yeah, thank you. And also, I'd like to thank Amy and also Provost Office members who are giving me this great opportunity. Today, I want to introduce myself and also my unit research to OIST community for future development. As you know, our body consists of a large number of cells. And based on this paper, adult human body consists of about 40 trillion of cells, which are roughly 5,000-fold larger than world population in 2026. It's so huge, a very large number. Also, amazingly, we were born as one cell. Please imagine one cell increase its number and cell type to make functional body during early development. This is totally amazing and beyond my imagination. Cell can increase its number by cell division. This is a cartoon showing a typical animal interphase cell. The nucleus contains DNA. DNA is very important because this contains biological information. We received this information from father and mother, and this is very precious and unique and determine your identity. Then, during interphase, DNA is replicated precisely. Then, during mitosis, this DNA is condensed and form this condensed chromosome. After nuclear embryo breakdown, microtubule polymer interact with chromosomes and align and segregate to daughter cell to maintain genetic information. This structure is called a mitotic spindle. And this only transiently formed during cell division process to segregate chromosomes. This is the current model of animal mitotic spindle. Spindle consists of a lot of short microtubules. And the microtube consists of alpha-beta tubulin dimers. And the tubulin dimer polymerize at the plus end microtubules and shows growth and shrink. And the minus end shows relatively less dynamics. And the minus end or microtube accumulate around this microtube organizing center, centrosome. Then grow microtubules and form the aster microtubules. This is called astral microtubules. Then plus end interact with cell cortex or plasma membrane. Also, plus end microtubule interact with chromosome at kinetochore. This connect chromosomes with spindle microtubules. And these microtubules are bundled by microtubule binding protein to stabilize this kinetochore microtubules. And this spindle is formed in all eukaryote and all developmental stage, including this one cell stage. After fertilization, the pronucleus migrate and form the first spindle, and our long journey starts. Okay, let's look at the actual example. This is a medaka 2-cell embryo showing a chromosome and microtubules. Like this, chromosomes are condensed and align the center of the spindle and beautifully segregated to daughter cell. It's very dynamic and beautiful process. Spindle ensures equal chromosome segregation, and abnormal spindle function causes chromosome missegregation, which leads to the gain or loss of chromosomes. This is called aneuploidy. And also sometimes chromosomes are damaged or clipped by cytokinetic machinery and abnormally distributed to daughter cell. Now, this is called micronuclei, and this promote massive chromosome rearrangement. Or recently, it is reported this is transported to other cell. And this is a list of showing the frequency of aneuploidy and also chromosome missegregation rate. As you can see here, yes, cancer cell shows very high frequency of chromosome missegregation. This is during mitosis, and this is a meiotic division, which generates sperm or oocyte. As you might know, yeah, human oocyte has relatively large number of missegregation rate. It depends on aging. Now, as a researcher extensively studied this, then now I'm curious here, this number. Human zygote also have relatively high number of chromosome missegregation rate. Now, several reports suggest zygotic division is error-prone in mammal or human. Initially, I expected early division should be accurate because if this is abnormal, downstream this effect is transmitted to downstream daughter cells. However, this is different. Okay. So for roughly 20 years, I'm interested in this mechanism, and I have studied spindle assembly and chromosome segregation. Also, we are studying spindle positioning because spindle positioning can determine the distribution of non-genetic information, such as daughter cell size and the location or polarity of cell. Cell division can be classified into two types, symmetric or asymmetric division. Symmetric division generates two identical daughter for clonal expansion, whereas asymmetric division generates two different types of daughter cell to increase cell type diversity. The balance is very important. So now we are interested in the mechanism of spindle assembly and spindle positioning in my unit. Okay, I slightly changed my topic. I want to briefly share my previous career development. Maybe I hope this will be useful for PhD student and postdoc. As Keiko mentioned, my academic career started at Kyoto University in Professor Mitsuhiro Yanagida's lab. In his lab, he used fission yeast to study the chromosome segregation. He identified a lot of mutant, temperature-sensitive mutant, showing chromosome arrest or cut phenotype, abnormal segregation, and also chromosome missegregation phenotype. As you know, fission yeast has only 3 chromosomes. That's why we can see chromosome unequal segregation just by looking at the mass of chromosomes, DNA. So this is a nice example to use the model organisms. Then he identified a lot of essential genes, many of which are conserved in human. Then my senior colleagues, Goshima-san, studied identify human mistlab, and Obuse sensei identified human mistlab binding partners using mass spectrometry. And I analyzed one of the human mistlab binding protein called brinkin, or now this is very famous as KNL1. Now we found this protein is important for kinetochore microtubular attachment and also mitotic checkpoint control. Then I joined Ian Cheeseman's lab. Here I shift my microscopic from kinetochore to the cell cortex to understand the mechanism of spindle positioning, especially spindle centering. To divide symmetrically, spindle is positioned in the center of the cell. The question is how spindle can find its center. Here we found two key internal gradient derived from chromosome and the centrosome. And these gradient control cortical dynein molecule. Dynein is a microtubule-based binding model. Then I'm happy to show our recent result about dynein later. Then I joined Professor Godo Goshima's lab as a non-PI assistant professor. However, he allowed me to establish my small independent group in his lab. And also Ian allowed me to continue my postdoc work. So I really appreciate these two supervisors. Then in Nagoya University, I want to introduce CRISPR-based genome editing. Now we can knock in these tags at specific target gene region to induce, to visualize or deplete or manipulate target protein. I want to achieve this. Then I collaborated with Professor Masato Kanemaki, professor at National Institute of Genetics. He developed excellent tool called AID, auxin-inducible degron. So we invited him last week, and he developed these technologies. Then we use two factors. One is MAID tag. The other is OSTR1. Both are plant protein. We use this plant protein in human or animal cell. This protein forms the SCFE E3 ubiquitin ligase in human cell. And in the presence of plant hormone auxin, this recognizes this tag and promotes polyubiquitination, which leads to proteasome-mediated protein degradation. Point is, protein degradation half-life is roughly 10 or 20 minutes. Very rapidly, we can degrade target protein. As you know, DNA is transcribed to mRNA. Then mRNA is translated to protein. And in many cases, protein has its own function. Previously, I used RNAi to analyze, to degrade mRNA, to analyze its function. However, RNAi target mRNA, but not protein. Therefore, if protein previously exists, we cannot degrade this. That's why I think this technique is provide very nice, clean result, and also very useful, especially in early embryo division research, because embryo has a lot of maternal protein. We need to degrade protein to understand its function. Okay. First, we use these technologies in tissue culture human cell, and we visualize dynein, and we found we successfully degrade dynein, and we found dynein is important for chromosome alignment. Then also we introduced manipulation. Here, I use light-inducible technologies. In short, we can target this protein called NUMA here. Then we found this is sufficient to recruit the dynein molecule and also to control spindle position in live cell. Now we established this technology. So I want to introduce this technology in live developmental context, for example, in embryonic cell. However, you know, human embryo is very, very difficult to use for ethical and also technical reasons. That's why, again, model organisms are very important. first candidate is mouse, but as you may know, mouse sperm does not provide functional centrosome. It's slightly different. Frog, excellent, especially for biochemistry, but not suitable for live imaging. That's why I was very curious to use fish. First, I thought, oh, fish cleavage is slightly different from frog or human, but maybe this is a problem of the cytokinesis, but not spindle assembly or chromosome segregation. But rather, this provides a very nice opportunity to find chromosome spindle on the same focal plane. That's why this provides a very nice opportunity to capture all spindle in early embryonic division. Okay. And fortunately, Professor Minoru Tanaka was in Nagoya University, and he studied medaka sex determination. Then, also, he already established a GFP alpha tubulin expressing medaka transgenic fish to visualize microtubules. Then he kindly allowed me to use this strain. Then I found, I analyzed, visualized this, and then I was very surprised to see, oh, the spindle structure is very different from tissue condition, human cell. And this orientation, nice figure. So I want planted and against now that I just think another question why that's why then in this video episode understand explanation. But first, yeah, of course, it might be good to use human embryo, but this is too complicated. That's why that's simple from my PhD work. And also the history in the trees I wanted always is very, very useful. And also this is best COVID pandemic. Yeah, that's kick distance from competitive or candidate first big competition. Also, I love diving mesh and genetic pattern. That's why I want to use live fish. That's why baby fish is too much will have that larger red one. Also, personal preference and the visual advantage is very important. For example, maybe you know, to measure fish model called one is medaka. There is that medaka is Japanese. This thing that I was assured is that also I found there are a lot of nice resource things. We should now we are in Japan, not in US. That's why we should take a condition. You might be interested in difference between medaka and zebrafish. This is similar, very similar, but some difference. For example, yeah. Medaka fish, medaka pair produce egg every day. You know, we collect egg today. Then tomorrow we can get egg from the same pair. This is very nice for experiment. Also, yeah, genome size is smaller than zebrafish. This facilitates genome editing. Also, permissive temperature is relatively wide. This is very attractive to me. Also, medaka has a lot of inbred lines, a lot of closely related species. That's why I feel a strong potential to medaka, especially for my research purpose. Of course, zebrafish is a very excellent model, but for us, maybe medaka would be better. And also, again, zebrafish is very popular. That's why I prefer using medaka fish. In addition, in Japan, medaka fish are used in elementary school in 5th grade science class to help students understand how animals pass on life. Like this. We compare human and fish. That's why I thought if we can get a nice result, it might be good for outreach activities. Okay, then let's move on. Scientific conclusion. Then we are interested in the mechanism of spindle assembly and positioning because these are important for to maintain genomic information and non-genetic information during development. However, we already know a lot of mechanisms and key factors, but they depend on somatic cell models. Also, we know early embryonic division called cleavage have very unique features. For example, large cell size. Also, transcription is off or suppressed, and the cell cycle is very rapid, SM, at least in fish and frog. And also, checkpoint is not functional, at least in medaka. However, chromosomes should be equally segregated. That's why I want to look. Then this is the first I just quickly check chromosome and the spindle in 2-cell stage and later stage. Then we found, look, chromosomes are almost similar, very similar, but microtube, spindle structure and size are very different. So this is the same embryo. Then they use the same tubulin protein. However, the assemblies are very different to segregate chromosomes. Based on this, we hypothesized mechanism of spindle assembly and positioning would be different between cleavage and somatic division. To understand that, first we tried to establish transgenic medaka line to visualize chromosome. Here we try to inject Cas9 protein guide and the donor construct coding M-cherry at RCC1 gene locus. This is chromosome marker. Then we finally get this knock-in string. Then after that, we crossed this EGFR alpha tubulin expressing medaka fish strain to visualize both chromosome and microtube. And this is the first medaka fish video showing chromosome microtube. And as I mentioned before, in fish, cell divide as the periphery of the egg. Then we invert it like this. Then we take the 70-second image. Then we show the stop view like this. Okay. Then we fit the egg in the channel made by agarose. Then we took the image using spinning disk confocal microscope. Okay. This is one of the highlights of my talk. Then this is a male pronucleus. This is a female pronucleus. Then after fertilization, they fused and form a nice, beautiful bipolar spindle, and they undergo repeated cell division process. It's very dynamic and also very beautiful. Now we took time-lapse imaging roughly for example, 10 hours. Of course, imaging causes damage, phototoxicity to the embryo. However, this embryo still continue normally the development. Then they can hatch. Therefore, now we believe we can capture the physiological dynamics of cell division process. This provides a lot of new information. For example, cell cycle is roughly 30 minutes. Then mitotic duration from nuclear embryo breakdown to anaphase onset is roughly 10 minutes. It's very fast. In tissue culture human cell, it takes roughly 30 minutes. But in this case, only 10 minutes. Medaka fish embryo can capture the 24 pairs of chromosomes within 10 minutes and nicely segregate without a clear error. Also, maybe you already know this. Nucleus is positioned in the center of the cell. Then divide again. The orientation is very nicely controlled to make symmetrical pattern. So we don't know why, but this is very exciting. And we already published this. Then you can, of course, download this. Then please use this under the license of CC BY4. Yeah. And recently we got this award, and I described some explanation. So if you are curious, please check this. Then we carefully analyzed spindle length during the development. Then we found dynamic structural change during early embryogenesis. Especially, I want to emphasize that medaka spindle has dense microtubule network at the spindle midplane around the chromosomes. This is specific in early embryo. And this seems to be stabilized. That's why I want to know these mechanisms. In the literature, other groups suggested RCC1. We use this as a chromosome marker, but this is actually enzyme to produce GTP-bound form of RAN. Then this RAN GTP dissociate inhibitory impulsion from spindle assembly factors.
Therefore, there's a chromosome that RAN GTP actually the skin color complex which stabilize or nucleate mutations by the gas. To test that, we want to regulate antisense protein by using this alternative system. To make a long story short,
yeah, we could establish RCC1 MID Clover flag homozygous knock-in strain. Then we injected mRNA encoding OST1 plus mCherry-H3B, which we can monitor the expression of OST1 by this red fluorescence. At the same time, we can monitor the chromosome dynamics. As you can see here, in response to the expression of mCherry fluorescence, now GFP fluorescence decrease, suggesting AID system work in medaka fish embryo. This is a movie. In response to the expression of the red signal, green signal is reduced. Then we could see very, very abnormal chromosome segregation phenotype. Something like that. Yeah. Then when I first look at this, oh, this is very similar to fission yeast cut a mutant. And then in those days, Professor Yanagida was still in OIST. That's why I print out this image and visited his office and showed that. Then he praised me. Yeah, when I was a PhD student, he barely praised me. But in this situation, yeah, he praised me. I was very happy. And after that, yeah, this caused embryonic lethality. If we look carefully, this shows several slightly different phenotype. Depends on the depletion level. It's slightly difficult to see, but yeah, it shows lagging chromosome or micronuclei or lagging or this non-disjunction phenotype. But in all cases, we also found abnormality of the spindle. Dense microchip network is not formed when we deplete RCC protein. So now we speculate this is one of the reasons to induce this abnormal segregation in early embryo. And now we can degrade RCC protein in later timing. If we use cell culture, we just degrade, that's it. But now we have developmental axis, right? Then we can, by controlling the timing of oxygen addition, we can degrade this target protein in later stage. Then we can analyze its phenotype, and we can compare. So yeah, we could degrade RCC1 in later stage, and interestingly, we couldn't see any chromosome misregulation phenotype, suggesting RCC1 is dispensable in late stage embryo. Of course, it caused slightly mimetic delay, and this caused additional defect after mitosis, especially nuclear envelope reformation is is very abnormal. But yeah, spindle assembly and chromosome segregation is basically normal. So this is a summary of this paper. First, I would like to thank these external contributors. And the point is, yeah, previously we and others found in small somatic cell, RCC1 is non-essential. Likely, centrosome-dependent nucleation is enough to make functional a spindle. But in large embryo, chromosome-derived RCC1 dependent longevity signal is essential, become essential to make a functional spindle. This situation is similar to oocyte. Okay. Then, now we established a basic strategy. We can establish knock-in strain for gene X and analyze its localization and depletion phenotypes in early and late stage embryo. Then, today I do not show the data, but we already succeeded in performing rescue experiment using wild type and the mutant after depletion of endogenous one to understand the domain function. Now we already established 1, 2, 3, 4, 5, at least 5 diagonal knock-in strain. Then one is now evaluating, and now our other member are trying to establish new strains. Let me briefly show our recent result about dyning. So, as I mentioned before, dyning is microtube dependent minus n directed motor. Dyning is highly conserved in eukaryote, and dyning transport almost all organelle toward minus end. That's why very important. Also, during mitosis, dyning is important for chromosome spindle attachment and also for focusing and also spindle positioning, at least in small somatic cell. I'm wondering where and how dyning works in extremely large embryo. This shows a medaka embryo and the human HCT116 cell with same scale. Now you realize how big it is. Then first we established dyning M-cherry knock-in strain and also dyning AID Clover knock-in strain. So here we visualize dyning using M-cherry together with EGF alpha tubulin. Dyning is shown in magenta. And yeah, please look. Did you see something interesting? We could see halo-like enrichment at the periphery in cytoplasm. Oh, the dyning shows a halo-like enrichment at the periphery in cytoplasm in metaphase. This is totally unexpected. I have never seen such cytoplasmic enrichment in smaller cell. And also in other organisms, for example, in C. elegans, as a group visualized endogenous dyning, but they have never seen such cytoplasmic enrichment, likely due to the size of the embryo. Okay. Then also we analyzed dynactin's localization. Dynactin is a dyning binding partner. They show a very similar localization pattern. Now we found this enrichment is well regulated during cell cycle. To understand its function, next we deplete dyning-dynactin interaction using this inhibitor called P150CC1. Then after injection, this disrupts dyning localization at centrosome or spindle or halo like this. This causes phenotype at metaphase, but the most unexpected phenotype was observed in anaphase. This is controlled. In anaphase, you can see large aster expansion, something like that. Right? However, when we deplete dyning, we could see a lot of microtubule nucleation in cytoplasm. this causes very abnormal cleavage like that. Maybe this is easier to understand. In normal situation, microtubule dominantly grow at the aster like this. However, when we inhibit dining, we could see a lot of feather-like microtubule nucleation in cytoplasm. I do not explain the detail. If you are curious, please check this bioarchive. Now, we already submitted this manuscript. Okay. This is a summary. So initially, we hypothesized mechanism of spindle assembly and positioning are different between early and late stage divisions. Now, we can provide strong evidence showing this. And in the future, we want to understand these mechanisms. And also, we want to know how this is remodeled during early embryonic divisions. Maybe you might want to know whether these mechanisms are conserved between medaka and other vertebrates. Of course, early cleavage patterns are very, very diverse between different organisms. However, for example, this is a recent report from other lab showing the human embryo in first stage and late stage. If we look this carefully, of course, microtubule probes are different between these experiments. That's why we should be very cautious. But this data suggests in early embryo, to me, microtubule is more dense near the chromosome compared to later stage, similar to embryo. That's why I think partially mechanism may be conserved. We want to continue our approach, and also in the future, we also want to introduce new technologies to manipulate spindle formation or spindle positioning using, for example, laser ablation or optogenetic technologies. Also, personally, I want to identify new genes essential for embryonic spindle assembly and positioning. Previously, we had a unit review. Then one reviewer said, our research is good, but we only target all the players. I agree. Yeah, as a first step, we should focus on all the players, established one. But I personally believe there are still unknown early embryo-specific essential genes. In the future, I want to identify this using several techniques such as mass spec. Then, if we could identify this new gene, I want to show that to that reviewer. Also, the same reviewer gave us very nice word, blue ocean. This might be the best word which I received in my research career. As you know, blue ocean means very pioneering and very, less competitive work. The opposite word is red ocean, very competitive. Of course, there are advantages and disadvantages, but I personally prefer a blue ocean. In the future, we want to get something. We want to discover something very unexpected and totally exciting finding. This is acknowledgement. I'd like to thank all previous and current members. As Keiko mentioned, we started our unit during the corona pandemic. Then after that, we gradually enjoyed Okinawan food and nice restaurant, something like that. Currently, our unit has 5 students. So far, I have not yet graduated any PhD student. That's why this is my one of the key goals in the next 5 years. I want to graduate PhD student. they already have a nice data. That's why I'm relatively optimistic. Also, yeah, this is very impressive pictures to me. They kindly celebrated my birthday. Oops, sorry. Then, yeah, I'm very happy to be able to work with these students and other members. I also want to thank former member at Naga University and the collaborators. And in OIST, recently we shared the journal club with Keiko, France, and Midori. And recently we co-organized this workshop. It was very nice. And Narita-san kindly synthesized several chemicals. And also, yeah, we'd like to thank especially animal resource section for medaka feeding. At the beginning, I and my wife came to the lab every weekends for feeding. But recently, yeah, they kindly helped that. It is very important for our research. I'd like to thank these organizations for grants. Okay. Then when I consider the tenure, of course, I want to thank all previous supervisor teachers. But especially, I'd like to thank these sensei, Shinshi in Okinawa was. Without their supervision or mentoring or support, maybe I couldn't get this current position. I really appreciate their support. Also, I want to express special thanks to Ai-san. She established a lot of knock-in strain and established live imaging and ALD system in medaka. So this is Okinawan's word, ippi nifi ribil. Thank you very much.
very much for the fantastic talk. We really enjoyed. So maybe we can take a couple questions. So any questions or comments?
Thank you. It was very nice story, especially establishing new system is a lot of work and it's impressive. I have two questions. The first question is, I was surprised medaka tolerate a very wide range of temperature from 4 degrees to 37-ish degrees. So is there any good temperature for embryogenesis, or embryogenesis also tolerate a whole range of temperature? So, yeah.
Yeah, good question. And I can show you this one first. Yeah, maybe this one, right? Okay. Yes, so this is the permissive temperature for adult fish. Then for embryo, the situation is slightly different. If I understand correctly, of course it depends on different species. Then roughly permissive temperature is 20 to 37 degrees for embryo.
Okay.
But this is very slightly different in different species. That's why, but this is interesting because if we compare different species, we can narrow down the essential genes required for to explain this permissive temperature.
Thank you. And the second question is, can you locally target dining? For example, you showed a really nice halo localization of dining. Can you specifically knock down that locally dining?
Locally knock down dining.
Yeah.
Locally knock down dining. Recently, other group reported optogenetically controlled dining inhibitor. Okay? Now, currently we are using P150cc1 fragment, but this can be